So this is my first session since the exam closed. These are my memes. I told you guys there would be memes. There are always memes. So I figured I would open it up to questions if you guys have questions about the test. I've had a couple of people email me questions about the test. And so I don't know if you guys had a question on the test that you got like wrong and you just have no idea why you got it wrong. Don't just be like, oh, well, I don't know why I got it wrong. The final exam is cumulative, right? So if you don't understand it now and you never figure it out, I promise you in December, you're not going to have it figured out because you don't just learn for no reason, right? So if you guys want to if you want, you don't have to, and we can just completely skip this portion. But you can go and open your exam, and if there's one that you're like, I have no idea why I got this wrong, or I want to know why I got this wrong, or whatever, we can talk about it. And then, yeah, so you can be scrolling through that, and even if halfway through the session, you're like, oh, I got this wrong on the test, just ask any questions you want, okay? Our biology fact for the day is that giraffes can live longer without water than camels. Giraffes can go 3 weeks without water. Camels can go without 2. Can go without water for 2 weeks. So that begs the question, why do people prefer camels over giraffes in the desert? One, it's a little bit trickier to ride a domesticated giraffe. The other is that this is without water. So camels can store water in their humps for weeks. Once they run out of that water, they can live for 2 weeks after. So giraffes go longer without water. Camels can store more water though. So I guess in a way camels go longer without taking a drink, I guess. But I thought that was interesting. All right, so before we jump into chapter 11, does anybody have any questions about anything biology related, be it from exam 1 stuff or exam 2 stuff?
So will Dr. Mott write the next, half of the next test?
Dr. Mott writes just however many questions his material covers. It probably won't even be, it'll be maybe a quarter of the next test because he only had, Friday and Monday. He only did 3 or 4 lectures that are exam 1 stuff. So most of it will be Dr. Pogorski stuff. Which brings up an interesting point. We have a new professor now. Not a new professor. We have a different professor now. Probably throws you for a loop to have somebody else up there. The other thing is he writes his tests differently, and he presents material differently. I don't know if you guys noticed he's got a different teaching style, obviously, than Dr. Mott, because everybody has their teaching style. Everybody always asks me who's better, who's worse. I don't get into that discussion at all because, A, that's not appropriate as the SI to be telling you if I have a favorite professor, which I do not. But everybody has their teaching style and everybody has their learning style, right? So for some people, Dr. Pogorski just makes more sense. And for some people, they're like, I don't understand what he's saying at all, right? I personally found Dr. Pogorski's material and testing style and all that to be more challenging, but in that sense I also learned it better because I was like, oh crap, he really pushes you harder. So it's also going to be up to you guys. Do you guys have questions about maybe Dr. Pogorski, his tests? I know that's kind of open. Someone has to ask stuff, or I'm just going to just stand up here.
From the content we learned today, what would one of the questions he might ask you?
If we get to the slides on signaling, I have a bunch of questions with it. So that would be the best time. But yeah, he words his questions differently than Dr. Mott does. And he focuses more on concepts than on just kind of rote memorization.
Will he do more questions like the party one today, where he rationalizing it?
Yeah, a little bit. He likes those kind of out there questions that if you don't understand his thought process and how he made the question, you just don't even know how to answer, right? So yeah, he does those a little bit. So that's just something to look out for. I don't know how to tell you besides he's more difficult for some. Other people are like, no, I totally get where he's going with this. But for me and for a lot of people, it's harder.
I think it was mentioned earlier in the semester that everything that was going to be on the test was going to be things from the lecture. Does that also stand for the course?
Yes, if it's not said in lecture, it won't appear on the test. So if it's in Mastering Bio and you're like, I have no idea, we haven't even talked about this in class, get it right on Mastering Bio because it still counts towards your grade, but it's not test material unless it's been said in class. For 1610 and 1620, regardless of professor, all across the board. Other questions? Should we just get into it? There's a lot of stuff today. All right, so the study tip is to maintain a constant sleep schedule every day of the week. Yes, even the weekend. And I know this sounds like something people say but don't do, but there's a lot of research behind it saying that if you get 8 hours of sleep a day, or 7 to 9, everybody kind of varies, that's good. But if you're not going to bed at about the same time and getting up at about the same time, you're still throwing off your sleep schedule. So even if you get 8 hours every night, but one night you go to bed at 10, the next night you go to bed at 2, the next night you go to bed at midnight, the next day at like 3 o'clock, you're completely destroying your sleep schedule, right? So you might think, oh, well, Tuesdays and Thursdays I don't have class till noon, so I can sleep in till 11, so I can stay up till 1 and doing all this, just go to bed and wake up at the same time. And your sleep schedule works out a lot smoother, and then your ability to learn goes a lot better, and then your ability to take tests and answer weird questions like Dr. Pogorski's is way better, okay? So just trust me on that one. Did we end up getting a roll going around? Is that happening? Thank you. So per the usual, 8:30 or 9:30. Every time, put it on the roll that you're supposed to go to class to, even if that's not when you go to class. There's always like 3 or 4 names that I type into the roll and they just don't show up. And then when I have to finish that whole 8:30 roll, I open the 9:30 roll and then their names start showing up. I don't care what time you do go to class. What time are you supposed to go to class? All right. So we're going to kind of breeze through photosynthesis because it's basically aerobic respiration with like a few minor differences, and in a lot of ways it just kind of does it backwards, but it's basically the same thing, right? So this is the electron transport chain and oxidative phosphorylation, right? This is still inside of mitochondria. So I wanted to compare it to, and you guys know anaerobic and aerobic fermentation. So anaerobic fermentation in animals creates lactic acid, in plants, ethanol and CO2, right? So photosynthesis, this is inside of a plant. They run photosynthesis. They get oxygen and sugar. They produce oxygen and sugar, right? They expel the oxygen into the atmosphere, and that's what we breathe. And then they put their sugars into their cells, and that's what we eat. And then they run aerobic respiration because they also have mitochondria, right? Don't get that confused. People think, okay, animals have mitochondria, plants have chloroplasts. That's true, but plants have chloroplasts and mitochondria, okay? So they can also do aerobic fermentation to make ATP. And just like aerobic fermentation in animals, they get H2O and CO2 out of it, and they actually use that, right? What do plants need to survive? Sunlight, soil, carbon dioxide, and water, right? Now, they get some CO2 also out of the atmosphere. They get lots of water out of their surroundings, right? But part of it they actually make kind of on their own, right? So that's pretty cool. Okay, so this is the whole photosynthesis in one go. I feel like I need to, I can't remember how I did it. That one. Makes it a little easier to see, right? So this is the whole thing in one go. So we put light into, what's that thing called? A thylakoid, right? Those are inside of a what? Inside of the chloroplast. So So we put light energy into these thylakoids, some water, and then we get out oxygen, ATP and NADPH. We put those into the Calvin cycle with CO2. We get NAD plus ADP and CHO2 or sugar. And so they kind of feed into each other. It's not like a self-sustaining system. That would be really nice because then plants wouldn't need any external anything. But it's just, it kind of feeds into itself. It still needs water and light and carbon dioxide from the atmosphere, but it can give itself some of the products back, okay? So this is a chloroplast, right? And this is its thylakoid membrane, and it's got proteins embedded in it, just like the mitochondria has. Just, we work backwards, and then this thylakoid space would be comparative to what part of a mitochondrion? The matrix, and the stroma would be like the cytoplasm. Yeah. So, question.
So what's the difference between stroma and cytoplasm?
Cytoplasm is made of cytosol. I don't know what stroma is made of, but obviously not cytosol. Anything made of cytosol would be cytoplasm. So stroma has just different ingredients. It serves the same purpose. It's just not called cytoplasm because it's not made of cytosol. You may have to just Google maybe what stroma is made of. That's also not something you're going to be tested on. So.
And then chloroplast and stroma, what's the difference between those?
Chloroplast is the whole thing. Stroma is just the fluid and stuff inside. Other questions? I know I'm going like mega fast. It's because I don't want to spend a lot of time on photosynthesis. So this is a photosystem, right? All of these are called antenna pigment molecules. And then this center one is a reaction center. And then this square one is a primary electron acceptor. The idea is that a photon of light comes in and gets kind of bounced toward the reaction center chlorophyll, where it's able to actually transfer the electrons over to the acceptor, right? This doesn't seem like the best way to do it, but you have to remember that photons are super unpredictable, right? The photon could land here, it could land here, it could land here, and they're all going to send it here, right? No matter where it lands. So the purpose of the antenna pigment is just to focus it into that acceptor the fastest way possible. Plus, the more antenna pigment you have, the more photons you can catch and channel into the acceptor, right? So just know where the antenna pigment are and what they do, what a reaction center chlorophyll is, this electron transfer, and that primary electron acceptor. Just know what they are and what they do. And it's pretty simple. That's about all you have to know there. Questions? As always, these slides are posted if you need them.
So is the electron from the photon?
Essentially, yes. The photon is energy, and we'll see it in a second when we get deeper into photosynthesis. But the photon is just energy. It's just light energy. By passing it down, then we're able to boost the electrons in energy level and pass them over. So the electrons aren't coming off of the photon per se, because photons are just photons. That's it. They're just energy, and they boost the electron.
Okay.
So it's kind of it's a two-piece deal there. Other questions? So what does this look similar to? The electron transport chain in mitochondria, right? That's because it is the electron transport chain in chloroplasts. And it works very similarly. I'm just going to point out the most important pieces, talk briefly about their function, and that's about it. Photosystem 2 and photosystem 1. And somebody leaned over to me in class and goes, who the heck decided to name them that way? It's just because they found photosystem 1 first, and they just called it photosystem. Then they found this one, and they were like, well, it's the exact same. We'll just call it photosystem 2. Then they realized it's actually in front of the other one. And then they were like, well, it's too late now. We can't do anything that would make logical sense. So we'll just leave it how it is. So yeah, photosystem 2 appears first. And then, so this is where a photon hits, right? These are your antenna pigments. it gets focused into the middle, okay? And then it boosts the electrons along the chain. These are just other proteins, okay? Not super important. It just gets boosted along its way, right? And we're able to split water. That's an important piece. We're able to pull hydrogens in. Remember in mitochondria, which way do they go? They go out. So in photosynthesis, it's the exact same thing. They just come in instead. So we build the gradient in here, right? Then we start getting pretty low in energy on our electron. So photosystem 1 takes more light energy, bounces it off the antenna pigment, focuses it in the middle, boosts the electron back up, and we get some more energy out of it. We end up with NADPH. And then with this proton gradient, same thing as last time. We push it through that ATP synthase, exactly the same mechanism as mitochondria, and we get ATP, right? I know I'm breezing through it. I hate to do it, but we have so much stuff to cover. So that is all the time I'm going to take on that, unless you guys have a question with that. You're just blindly trusting that I've given you everything you need to know. I heard somebody say always, and that touched me. It's also a dumb idea. Noncyclic electron flow, okay, that's one way to run these two photosystems, right? Same thing as we've just been seeing. Light enters, bounces off, gets focused, goes up, loses energy, gets bounced up again, and goes to the end of the cycle. Like I just showed you in the last slide, right? Non-cyclic, meaning it's basically a line, right? It's not a straight line, but it doesn't come back on itself at any point. There's no cycle back. It just goes straight through, okay? We get NADPH and ATP, right? Cyclic electron flow, on the other hand, focuses primarily right here, okay? So we boost the electron up, it comes down, it loses energy, it gets boosted up again, but we pass it back into the cytochrome complex, come back down, boost back up. I don't know how many times it does this. You don't really need to know how many times it does this. For all I know, it does it 1,000,000 times or one time. Doesn't matter. It's just the idea that it goes back on itself, comes back in, gets boosted up, and then goes the rest of the way through. So the cyclic part is that right there, okay?
So when it goes back on itself, is it sending anything to produce NADPH? Or is it just going in a circle and just wasting time?
It's not so much, it's not wasting time. We're getting more product out of it. It's just, it's going to run the cycle again, not to do nothing, but to make more product. And we'll see it in a second. But it's not like most of it goes here and part of it goes there. The whole thing does another cycle and then moves on, all as one unit. Other questions? Oh, way in the back.
So like for the mitochondria, we have to remember like how many ATP were produced per glucose molecule. Is there any like amount of ATP we're gonna have to remember is produced here?
Not as far as I'm aware, and I don't remember it being mentioned in class. Does anybody remember like a specific ATP number being talked about in class? I don't either. So unless I'm wrong, it wasn't mentioned in class, so it shouldn't be something we worry about. Other questions? All right. So this is only in photosystem 1, right? As I pointed out, this cycle is right here. We get ATP out of our proton gradient. We don't get any NADPH because, again, we're just focusing on right here. It'll eventually go on, but for right now, we're just talking about the cycle. And we don't split water, because where do we split water? Photosystem 2. That already happened, okay? So know those differences. Know non-cyclic versus cyclic, where they occur, and then know that cyclic has no NADPH and no water splitting. Okay? Questions? All right, moving on then to today's material, signaling, right? This is where Dr. Pogorski comes in. So everything up to here, Dr. Mott himself will write the test questions. And then from here on out, the rest will be written by Dr. Pogorski. And I would kind of roughly guesstimate that to be like 3/4 of the test ends up being Dr. Pogorski's. I don't really remember. It may come up in class. You're also always welcome to ask him yourself. He'll know. Okay, so 2 types of signaling, intercellular and intracellular. Okay, by their prefix, you should be able to know where they occur, inside and outside of the cell. Intercellular is the real kicker that has 2 different kinds, and those kinds each have subcategories, right? So there's direct contact with cell junctions or cell-to-cell recognition, also called contact signaling. You'll see it either way. Then diffusible signals have paracrine, synaptic, and endocrine, right? So this is where he kind of did like the whole party, and did he do the telephone line comparison, or did he just mention the party? He does it different every year, and I forget which way he does it. So we're going to get into each of these. Intracellular signaling, not as bad, right? There's just intracellular signaling. All right, so based on these images here, try and figure out what kind of signaling is going on. First of all, freebie, intercellular or intracellular signaling? Intercellular signaling should be easy enough, because if there's 2 options, then it has to be intercellular signaling, since there's only one kind of intra. So take a second and try and figure out, first, is this direct contact or diffusible? Then is it one of these 2, or is it one of these 3? Okay, so take 20 seconds to look at that. Which kind is it? Do, 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 do. Okay, what do you guys think A is? Gotta say it loud and proud. Cell junctions. Why? Because they actually join, right, to each other, and they send their signals through their cell walls or their plasma membrane, depending on what you're talking about, to each other, right? So then B would be cell-to-cell recognition, right? So those are both kinds of what? Direct contact, right? Cell junctions and cell-to-cell recognition. So they're the same idea. The only real difference would be that right here, see how that's the plasma membrane? See how the plasma membranes don't actually touch each other, but proteins sticking off of their plasma membranes do touch each other, right? So they are directly contacting each other. It's just not a junction. They're not touching cell wall or plasma membrane to each other. Okay? Questions there? We're doing decent on time. We're halfway out of time. All right, so then these 3 are going to fall under what category? Diffusible, okay. So we have 3 options now, because you guys remember there was 3 kinds. Take a second to try and figure out which one's which. And then I'm going to ask for comparisons, so the whole like talking at a party. Come up with your own, okay? Like a telephone line, you come up with your own, because I'm not very good at that, apparently. All right, so what's this first one? Oh.
Just really quick, what were the 3 different types of diffusible signals again?
Paracrine, synaptic, endocrine. So pick out of those 3, assign those 3 to each of these 3, okay? So that first one on the left there, what is it? Paracrine signaling. What could that be compared to? It's like talking at a party. Why?
You said why?
Yeah.
Because you have to be close. And like, I honestly didn't get it, because when you said talking at a party, I thought of the junction, because they were together and going back and forth, and this was only going out. So I didn't understand that one for sure, but you just said because they're going out, close.
Yeah. Do you have a question, a comment? Yeah.
So isn't it like a party because like there's multiple around?
Yeah. So if we consider this the party, right, you guys can all hear me because you're close enough. but people like in the next room over aren't close enough, they can't hear me. So paracrine signaling, those target cells there are close enough to receive the signal, but a cell on the other side of the body isn't going to receive that signal because it's not close enough. So if you're at a big party, the 10 people around you might be able to hear you, but somebody like at the top of the staircase isn't going to hear what you're saying because it's a big party and it's loud, right? So that's where he's going with that. Did you have a question as well?
Well,
Or comment?
I was thinking it made me think of like a therapist in a group therapy or like couples therapy.
Absolutely, yeah. So the surrounding, if you have group therapy and you sit your therapist right in the middle and everybody's in a circle and they say, hi, I'm Todd, hi, Todd. The people in the circle are receiving the signal, right? They can hear the therapist and they can hear like the people in the circle. But if, let's say you're holding it in like a big center, the people in the next room over having their own therapy session don't hear what's going on in this one, right? So paracrine signaling is just for the nearby molecule or cells, but they don't actually have to touch each other.
Is synaptic signaling only with neurons?
Yeah. As far as I'm aware, and as far as I think anyone knows, we may someday find something, but right now we've only found this in neurons because they're the only ones technically with a synapse in this sense. A synapse just means like a small gap, but it's talking about like the space between neurons. Yeah.
Didn't he say like you'd go from like neuron to muscle though? Like that way? Like that'd be synaptic?
Yeah, it's synaptic signaling, but it's because it's traveling down the neurons and then it gets to the muscle, and the muscle doesn't like continue the signal as a synaptic signal, if that makes sense. Anyway, so it sounds like we're ready to go on. What's I think this is the next one that's going to uncover itself. What's that one? Endocrine, if you wanted to cheat, it's literally right there. Probably should have covered that up. So what could this be compared to as like a real world example?
Someone with like a megaphone in a large crowd.
Megaphone in a large crowd, kind of. That might kind of confuse itself with paracrine though, because nearby people still hear it.
Maybe email?
Email, why?
Just 'cause if you're emailing one person, it doesn't matter the distance.
Yeah. So it'll go from this endocrine cell through your entire bloodstream, and it can go across your whole body and hit a very specific cell, right? Like an email. It can travel across the entire globe and go to one specific person. Or a phone call, right? You call a person, and we don't really call, it's a text message, right? It just goes straight to one cell phone, right? Nobody calls anybody anymore.
So say like food dye in a cup of water because it affects can affect the whole body.
It's tricky with that one too, because that kind of, you might confuse that with paracrine signaling because it would also affect the nearby molecules. Endocrine signaling specifically long distance. It's only long distance. So if you like put a PVC pipe in the water and hit the dye so that it just went to like the bottom or something, I guess, kind of like that. But it can't hit the nearby at all. So again, when you make up your own examples, do be careful not to maybe like make one that qualifies for both, because these are exclusive. Right? If it hits the nearby cells, it's paracrine. If it hits long distance cells, it's endocrine. And then if it goes between neurons, it's synapse, right? So that one kind of makes that obvious. Questions with endocrine before we start talking about synaptic? Okay, so synaptic signaling, I already said it a couple of times. Where does it happen? Between neurons, right? That's what a synapse is the gap between the terminal bud or the very end of a neuron and then the dendrite or the tips of the next neuron, right? So a neuron kind of looks like, it's a very simplified version, but it looks like this, kind of. These are the dendrites. This is the axon. And then at the very end, it has some terminal buds. This is a terminal bud. This is the tip of another dendrite, so like another dendrite's sticking off there. Okay? The synapse is that gap there between the end of one neuron and the start of the next. And synaptic signaling means that some chemicals pour out of the end of one neuron, pass through like a almost actually a microscopic distance, and then go into the next neuron, right? So this is where things like dopamine and serotonin and all those neurotransmitters move. That's why they're called neurotransmitters. They're inside of neurons, right? So if you take like an antidepressant, there's one called selective serotonin reuptake inhibitors, right? If you guys have taken like a psychology course, you'll recognize SSRIs. They just keep the serotonin from going back into the first neuron, right? And so they just kind of hang out in the synapse and they get absorbed a little bit more quickly. So some people with depression, the issue is too much serotonin. Well, normal amounts of serotonin comes out, but then it gets taken back up because you don't want just free-floating serotonin. Then things get all out of whack, right? But their issue sometimes is that too much serotonin gets taken back up before it can get absorbed into the next neuron, right? And serotonin's what gives you that good mood. And so they're not getting enough serotonin, and that causes the depression. So you take an SSRI, not as much serotonin gets taken back up, it hangs out, you get more, and your depression lessens or can even go away, okay? So whenever you take any kind of antidepressant, they're working on your brain, right, and the neurotransmitters there. Questions on any of this? I know I made a whole mess out of these slides. There used to be a way to there we go. Questions here? No? Is that roll still going around? Who has the roll? Awesome. It hasn't come to the front yet though, has it? Send it this way now. It started where, did it start here in the middle? So like this whole row here, did you guys get it? Okay, so let's start it here in, what is it, Juneau, Alaska shirt?
Yeah.
Are you from Alaska?
Nope. My parents brought it back for
Dude,
vacation.
nice. Alaska's sick. Anyway, it is. Alaska's dope. All right, so now let's talk about this process here. It's a 3-step process. We have reception, transduction, and response. Somebody quickly tell me what reception means. It's not what you do after you go to the temple. What is it?
Receiving the signal molecule.
Okay, perfect. That is it. The signal outside. Did he tell? Have we gone over the vocab word of what a signal molecule is? What is it? A ligand. Some people say ligand, which I think sounds really weird. A ligand attaches to the receptor protein that's embedded in the plasma membrane there. Okay, and that's it. Reception ends there. Okay, it just attaches to the protein, and that's it. What's transduction then? A lot of people are mumbling it very softly. I can't understand what you're saying. Somebody raise a hand and just go for it. No fear. There's no wrong answers. Well, there are, but.
It just passes the signal throughout the cell.
Yeah. So it's just sending the signal along. But why? What's happening here? Why is it going from like an orb to like a weird 80s chair to like a football? It's changing shape. Why?
So it can interact with the other proteins inside.
Okay, so it can interact with the other proteins. One of the unifying themes of biology, one of the biggest deals, a phrase you probably ought to memorize is shape determines function. Have you heard that in class a couple of times now? Shape determines function, always. So the shape of this relay molecule determines the signal that we get, right? So let's say we're trying to send a signal to flex our arm, right? And this football one is the signal that says flex your arm. Let's say that 80s chair one says flex your leg, and the circle one says blink your eyes, right? We're trying to flex our arm. This isn't a perfect example because that would obviously happen in your neurons, right? This isn't inside a neuron, but you get what I'm saying. So we want to flex our arm. We have to change the shape of the molecule till it's the shape that says flex your arm, right? This would probably be something more like activate the lysosomes in the cell, right? And this one says like activate the peroxisomes, and this one says like activate the flagella, I don't know, right? So we have to end up at the signal we actually want to have, because after that we get the response, right? Anybody not guess what the response is? We do whatever the signal told us to, right? We activate the lysosomes, we flex our arm, whatever response we're trying to get, okay?
And did he go over all three of these today? Because it felt like he just like went over reception.
He may have only touched on reception, so you may have just gotten a sneak peek, and if he contradicts me in class, go with what he says. But that's pretty basic. We will go into more depth later on in class.
So does the ligand pass through the receptor? Or
No.
does it stop there?
Except for steroids, I think, and we'll go over that too, so if I'm wrong, he'll correct me. But steroids are the only ones that will pass through the membrane. Everything else attaches to the receptor and stays outside.
So the transduction is the changing of the receptor.
That's what transduction means, is to change shape.
So it's
Not changing the receptor, though. It's changing the molecule inside that has received the signal. The receptor is this thing. It doesn't change shape in this process. The signal inside does. Does that make sense? And again, that also we go into way more depth in the next couple of lectures here. So if you're like, I don't understand one of those three or all of those three, we're still working on it in class. I just wanted to, I guess, get ahead of the curve on that one, and I just kind of skimmed over it. We'll get into it later. Other questions? Now you're like, I'll just wait till Pogorski says it in class so I actually understand.
So is it a molecule or is it like a protein?
It's a protein, yeah, inside that's getting changed. All right. Did he show you these? I feel like I'm showing you slides that he hasn't shown you. There's two kinds of reception, right? There's indoor and outdoor, but indoor is more expensive because the venue costs a lot. You have to cater. Come on. That's a decent one. It's better than Dr. Mott's puns, right? He's gone now, so I can say it. No, I'm just kidding. I like puns. I thought they were funny. Okay, so we have two different kinds of reception. Okay, so our ligands bind to the signal receptors. Okay, it's a signal molecule binding to a protein. So what is this one on the left? What kind of reception is that? Somebody said intracellular reception, okay, because this hormone, oh, so it's hormones, not steroids. I already corrected myself. Hormones can pass through the plasma membrane, and they go straight into the cell, and they conduct their signal straight to the nucleus on their own, right? They just dive right in. They do attach to a protein, right? They have to, but that protein is already inside the cell, intracellular. Okay? So what would you guess the other one is then? Cell surface. A lot of people say extracellular. It is extracellular signaling, but it also passes into the cell, so they try not to call it extracellular signaling. They try to call it cell surface reception because it's still going to go in. It's just that it's received at the surface of the cell, right? So the example from last slide with the transduction and all that, that's a type of cell surface. It's a protein embedded in the membrane. Ligand attaches still outside the membrane, and then the signal gets passed in, but the ligand itself never enters, okay? You guys see the difference there? As long as that difference is clear, you should be fine. You're not going to be asked any crazy questions on this, just which one is which, right? Okay, he covered these in class, the big ideas in reception, okay? So I already said it, shape determines function. So it's important that the binding of the signal changes the shape, right? That's how the signal happens. If the protein didn't change shape, then it wouldn't change function and nothing would happen. So it has to bind, change shape, get a reaction, right? Then that makes the receptor active in signaling, okay? And these are actually very specific, right? Not just any ligand sticks onto any receptor and makes signals happen, right? It's very specific, kind of compared sometimes to like a lock and key, right? It's got to be the right lock and it's got to be the right key. Otherwise, you can't get in, right? But why would we do that? Now we have to have more receptors and more ligands. Why don't we just have one master ligand and one master receptor?
Like that way there could be more like diseases or issues with the body with them not working.
Yeah. You could go to like jump over a log and you could poop yourself. Because it's just, there's no direction, there's nobody in charge, there's just one signal. You guys are slowly catching up. You were like, what did he say? Poop myself. You could, if it were just like one receptor and one ligand and they all just started sticking on wherever they want, you would do things uncontrollably, right? You would just drop on the floor and roll around for no reason because ligands and receptors would just boom, 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 boom. And you just get whatever happens to happen, right? So we have to have very specific ligand receptor binding so that you get the right reaction, the right response for the right situation, right? So now you guys are always going to remember. You're going to be like, oh, now I know why we have very specific, so I don't poop myself. I'm telling you, these things help you remember. I know you laugh and you think I'm a weirdo and I shouldn't be in charge of any of what's going on up here. It helps you to remember, okay? Come up with weird, funny, gross, whatever. Don't tell me, because I don't want to know. I tell you because I have a microphone. Keep it to yourself, but come up with weird, wacky ways to remember stuff, okay? It really helps. Come up with weird mnemonics. I had a friend in class, when I took this class, he'd make up acronyms, and they were always super inappropriate, so I won't tell you what they were, but he would come up with like super weird acronyms, right? And I'd be like, that's nasty, dude. Like, I don't want to know anything about that. And then I'd go and I'd sit in on the test and I'd be like, he kept making up that acronym about this, this, and this. And I'm like, as gross as it is, I know exactly how to answer this question now because there was a weird acronym for it, okay? Do whatever you got to do. All right. Intracellular reception, okay? The receptor bound to the hormone moves to the nucleus. The receptor hormone pair acts as a transcription factor, binds to specific DNA sequences, turns genes on or off. Does anybody recognize that nightmare of a sentence? From where? From class, right? This is how Dr. Pogorsky teaches. This does not make sense to me. Whether it makes sense to you or not, that's fine. But for those like me who are like, I don't know what's going on, break it down, okay? So you'll be like, what? I don't get what he's trying to say. Maybe draw it out, maybe make it into a story, do whatever you got to do, but break this down into pieces to actually understand what it is instead of going receptor bound hormone nucleus pair X binding to I think I know what he's saying. You don't. And you know you don't, but you're like, I don't want to. That's a lot of work. Do it anyway. Okay, so let's say this is our receptor. That's our first keyword there. What should I draw next? A hormone or a ligand bound to it, right? They're connected to each other. It looks like a UFO. It moves to the nucleus. So let's say this is our nucleus. It goes into the nucleus. It acts as a transcription factor. Okay, that's how this happens, is a transcription factor. And it binds to specific DNA sequences, right? Our DNA is in the nucleus. It binds to the DNA and turns the genes on or off, okay? That's the whole purpose of intracellular reception. Hormone passes in, binds to a protein, enters the nucleus where our DNA is, attaches to the DNA, and turns the gene on or off, okay? Name a hormone. Testosterone, right? That's pretty much usually the first one to come to mind, testosterone or estrogen, right? Because that's basically the big difference between the genders comes down to testosterone or estrogen, right? So what does testosterone do? And I know it gives you male characteristics such as facial hair, baldness. Who said it? You looking at me? I'm only 22, man. Freaking A. You have a point. Broader shoulders generally, right? Deeper voice, more body hair, those kinds of things, right? Estrogen, on the other hand, is where you get your female characteristics, usually wider set hips, not as deep of a voice, less body hair, all that stuff, right? And obviously it plays its roles in production, right? We generate sperm based on testosterone, generate eggs based on estrogen, right? All those things. Those are hormones. So those are involved in intracellular reception. So how does testosterone work? Now that you know how intracellular reception works, somebody walk me through what would happen. This is the level that you need to know for Pogorsky's tests.
So it enters into the cell, binds with the specific receptor, and then they both move to the nucleus to the DNA sequence that determines hair growth.
Okay, yeah.
And it turns it on for more, or turns it off. Estrogen would maybe turn it off.
Perfect. So everybody's DNA is pretty much the same. We have slight differences, right? That's how we end up taller or shorter or whatever. But we all have the code for body hair, right? Because we all have body hair. Well, with some slight exceptions for what's that called? Alopecia, where you can't grow body hair. That's a different thing. Then it's not testosterone, estrogen. But testosterone goes in, binds to your DNA, and turns on the hair growth genes, right? And keeps them on for longer, okay? And that's how you end up with more facial hair, more body hair, right? And with women, they have a little bit of testosterone. Women have testosterone, men have estrogen, right? It's just different amounts. Women, the estrogen goes in and activates all of its, related characteristics, broader hips, breasts, all that. But then it also has a little bit of testosterone that turns on the hair growth, just not as much, so you don't get as much body hair, right? So be able to take this and go that far with it. Go all the way down to real life examples with real scenarios, okay? So be able to see that and go, okay, well, how would this work with testosterone and estrogen that are like the most common hormones that we know? You don't have to be like a super advanced biologist to figure out just the basics of testosterone, right? You don't need to know more than that, but be able to turn it into a real life thing, because Dr. Pogorsky loves those where you have to apply it to the real world or take like a theoretical situation. He'll say, well, what would happen if intracellular reception stopped working? How would that affect body hair? And you might go, intracellular reception and body hair? I don't understand. If you know that intracellular reception is hormones getting into the nucleus, then you know the amount of body hair is going to decrease if we turn off intracellular reception, right? You guys see where I'm going with that? Hopefully. You'll learn it on the way.
So wouldn't DNA dictate hormones though?
Your DNA does also dictate the hormones, so it comes back on itself, right? Your DNA decides whether or not you have testosterone or estrogen. But then the estrogen and the testosterone determines whether or not you turn on other genes to have a deeper voice, more body hair, broader shoulders, whatever. Does that make sense? So it kind of feeds on itself a little bit. Other questions? Yes, I've been about this bald for like the last 3 years. I don't know why. I don't know what happened. All my parents, all my grandparents, everybody's got hair. I still do, but it's just embarrassing. All right. We're about out of time here, so I think I have a few more slides. Just remember that the cell surface, okay, the ligand never enters the cell, and then that shape change is what triggers transduction. Okay, the protein that the ligand just bound to changes shape, and that's what starts that transduction where it went from a circle to an 80s chair to a football, right? That change in the protein started that whole thing. I think we're getting into transduction more in class next couple of times. If we don't, I'll try and go over it more, but I'm fairly certain we're going to get into a lot more detail in the next few lectures here. Does anybody have any questions before you bail out? If you do, you can always just come up and ask or email me or do whatever.